So when we look at things, let's say just in a cell, a human cell in a petri plate, we don't get data on how that cell is interacting with other cells necessarily, or interacting with other cells across other tissues. By looking at a C. elegans in a petri plate, we actually start from the phenotype, and in this case, as I think you and I are interested in, it's the actual longevity of the animal itself. And so that gives us a great starting point to then look at further development.
Hi everyone, welcome to episode 9 of the Polling Principle. And I'm here today with Ben Blue, CTO of Aura. And I'm actually very excited to have this conversation. We met, I think, a couple of years ago, maybe something like that.
Uh,
And then
3 years ago, yeah.
3 years ago, it feels like a long, long time. And then we met again at the Pacific Northwest Life Sciences Conference. I think a few weeks back. And thank you for coming to the podcast. I think this will be a very, very interesting chat. So, to begin with, I'd love to, talk a little bit about your your path, right, to I guess to longevity, to biotech, and so on. And for the sake of telling everybody else that is listening.
Yeah, I'd love to, and thank you so much for having me on. So, as everyone here knows, I'm Ben Blue. So, I was born in a little fishing town up in, South Central Alaska, um, where the only biology performed was fisheries biology, so I never dreamed I would actually go into biology as a kid. But I was lucky enough to have, introduction to aging very early on. As, I was, raised predominantly by a single mother, and I was lucky enough to have the support of her mother and her mother's mother, my great-grandmother, all in one town and one house. And so even early on, looking at the different rates of aging and the different outcomes that people experience, even with very similar genetic background, was something that I was thinking about a lot. And so, uh after leaving Alaska to come down to Oregon to get my bachelor's degree, I really fell in love with what I found to be laboratory biology, realizing I could do biology indoors out of the rain. Um, and I was lucky enough to start working for Patrick Phillips, who studies the evolutionary biology of aging. And that's where I got started working with the model system I still use today, C. elegans. It was also where I started to help develop laboratory automation tools. So at that point, we were building very old versions of some of the technologies we use today. Um, After that, I came up here to University of Washington to study with Dr. Matt Kaberlein for my PhD. That's where we developed the Wormbot system and the Wormbot AI software pipeline that we now use at Aura to accelerate drug discovery.
Cool. Awesome, thank you. So let me go through a lot of the some of the things that you touched on
Yeah.
maybe one by one. So you mentioned evolutionary biology, right? And I think maybe longevity is one of those things that is not 100% well understood in the context of evolution, right? Like, I guess, what we see today is, maybe bigger animals like them to live longer, right? But it's not like a direct correlation, right? There are animals that are maybe evolutionarily very old, like sharks, right? That live
Mhm.
hundreds of years. There are lots of animals that are, evolutionarily more, I guess, novel, right? Like humans, we live, longer than we would normally would for our size, I guess. So, you know, what do we know, I guess, in terms of like, what is the impact of evolution in longevity, right? Is it evolutionarily advantageous to live longer or not, right? Like, what, like, how would you qualify that?
Yeah, so one of the big and I want to emphasize that there's much better evolutionary biologists out there than me. But
Mm.
one of the things that I think about when I think about the evolutionary biology of aging, evolution occurs at the point most 99% of the time at reproduction. So we end up seeing the selection for novel traits occur at the age of reproduction. So humans, that's anywhere from you know, early teens through early 40s. After that, there isn't a strong evolutionary pressure to drive genes associated with aging.
Mm.
There is some really cool work out there,
For females,
hypothesis
right?
for females. Right.
Yeah,
Yeah.
yeah, yeah.
Um, and even then, there is you know interesting sexual selection around males. One of the couple of places where telomerase is active, the enzyme that extends telomeres, is in the human testes. So uh, the gametes from older males actually sometimes have longer telomeres than those of younger males, but they also have a whole host of other potential hazardous somatic mutations that have accumulated over time. So there is kind of even there a trade off. Um, there is some interesting work looking at some of the social structures. So the grandmother hypothesis. You live longer, and that gives your grandchildren or even great grandchildren, in my case, a better chance of success.
Yeah. But I mean, I guess there's still a lot that we don't understand yet, right? Like because if reproductive capability somehow ends at age 40 something, but lifespan in average humans could be twice as that, right? So I guess it's a long period of time to be a grandmother, I guess.
Yeah, and actually, one of the things that, like in a lot of medicine, the effects of aging biology on women are still understudied. And when we think about aging, one of the most strongly age-associated phenotypes that's shared by 50% of the population is reproductive aging for women into going even into menopause. And it's really understudied, and there's some really, I think a lot of room to explore, especially as we think, as we talk later on about what sort of endpoints and therapies there are for longevity interventions. It's a really, really um, strong opportunity to have a lot of beneficial impact for a lot of people.
Hmm. Okay. So now maybe switching gears a little bit, right? On the next thing that you mentioned was C. elegans, right? Which is
Yeah.
a small nematode worm, right?
Mhm.
Like that's the name of it. And maybe at a high level, right? Like, um why do we do experiments on animals, right? Like, why do we learn, from those animals as opposed to doing experiments on, you know, human cells, right? Like that we take out of people or something else, right? Uh like what is the rationale for using C. elegans?
Yeah. So I think that's a great way to set it up. And I'll work down from even the high levels. Like, why do we work on animals? And, I think the short answer, people age as much as I wish we age slower, we still age slowly in terms of a scientific study. It's really hard to do those. And, there's a work going on to develop biomarkers of aging and clocks to let us assess endpoints in shorter term for people, but those really haven't hit the mainstream yet. So how do we look at aging? We look at, the gold standard is inevitably mouse work, right? Laboratory mice are really well understood. Even then, they live over 3 years. So we're looking at something that, maybe a grad student, if this is where I'm talking academics, maybe a grad student gets one full lifespan done. Sometimes even it's one grad student starts and another student finishes it. So what can we do that's even shorter? And so people have been looking at invertebrate animals for since about the 1950s. So I want to say 1954, some of the earliest work on C. elegans was done, and that was even prior to Sidney Brenner. Uh, these were establishing C. elegans as a genetic model. So, you know, at a very fundamental level, a lot of the proteins that govern C. elegans biology also are directly correlated with the proteins that are governing our biology. And so early genetic insights into human biology and how proteins work, how proteins are expressed, came from C. elegans. Now, they are a worm with about 1,000 somatic cells. So there's a vast world of difference
Oh,
between,
it's only 1,000 cells. I didn't realize it was so few.
yeah,
Wow.
1,000 cells, but a little over 300 neurons. So, and, we've gone and C. elegans researchers have mapped out the full uh, pathway of cell differentiation, starting with a single uh, oocyte, single celled egg. Now, what the next question that I often get asked is why can we why can we use this as a model? And so, even some of the earliest work into the biology of aging. So stuff looking at insulin signaling by Cynthia Kenyon, that was done in C. elegans. And so we've seen really strong effects going through there. Um, and the real, I think, strength of the model is that these animals only live about 20 days. And so when we think about the volume of compounds or interactions that we want to look at, this lets us do things on a scale no one else can really do. And short of some of the, you know, doing stuff on, 1,000 petri dishes, each with a human cell in it. But that doesn't ever necessarily tell you as much as looking at a whole animal. So, you know, I like to think that by starting with the phenotype and starting with, let me start this sentence one more time, if that's okay, Javier. Am I rambling too much?
Not at all. Mm.
Okay. So, uh, biology is fundamentally the most chaotic, complex system that I think anyone could ever look at. And so the level of interactions between within organelles within a cell, between cells in a tissue, and between tissues in a body are incredibly complex. And we don't really yet have the power to fully understand all of that. So when we look at things, let's say just in a cell, a human cell in a petri plate, we don't get data on how that cell is interacting with other cells necessarily, or interacting with other cells across the other tissues. By looking at a C. elegans in a petri plate, we actually start from the phenotype. And in this case, as I think you and I are interested in, it's the actual longevity of the animal itself. And so that gives us a great starting point to then look at further development. I was listening to some of your past episodes, and you talked about how uh, the journey of people trying to develop Alzheimer's disease medications has been really, really fraught by false signals. So many drugs, roughly what, 80% of drugs fail in clinical trials.
More than that, yeah, yeah. I think it's
More
about
than that,
uh, well, so it depends where you start counting, right?
sure,
Like you
yeah,
start counting at like lead candidates, right? So just speaking, if you start counting at drugs for which there is already an investigation on new drug and that they
yeah,
already started the clinic. I think out of those, yeah, maybe like 20% ended up succeeding all the way at phase three.
yeah. And so we think that we can enrich for a better selection of hits by starting with whole animals.
Yeah. So, I mean, I think that is very interesting, right? I guess there's a lot of maybe common biology that we share with worms,
Mhm.
like you said, the nutrient sensing and so on. And I guess the interesting thing about worms is like, they have different tissues, right? I mean, I don't know if they have vasculature, for example, but they do have neurons, right? And they have
Yeah.
muscles, I guess, right? Um, so, if, something affects a cell, I guess, and that ends up triggering some signals to other cells or whatever, like you would see it in a worm and you would never see that in a petri dish, right? Um, and I guess, but at the same time, I think the interesting question would be, well, can we use this to investigate aging, right? And I think, one of the questions that or one of the things that I've heard that a few people say about mice in particular is that a lot of mice would end up dying because of cancer, right? So
Mhm.
like the mice, for whatever reason, they have like I don't know, like a poor DNA replication mechanisms, so they end up developing cancer, so they die of cancer. So if you go and cure cancer on a mice, you can make the mice live longer. But the fact that you can make a mouse live longer doesn't mean that you can make a human live longer with the same mechanism, right?
Right.
So I guess the question is, it's a long introduction, but the question is, well, what kills nematodes, right? And like, how do we know that when we try different things on worms, right? Like, that actually not affecting, the cause of death of nematodes, right?
Yeah, that's a great point, and people have been looking into it. There's studies out there that investigates. One of the most common causes of death in C. elegans is actually colonization by the bacteria that they live on. So they lose the ability to break down the E. coli that they're commonly fed in a lab setting, and that breakdown eventually leads to the E. coli proliferating in their body and essentially running rampant as an infection.
Mhm.
Uh, the the thing that I think is useful is that the age-associated breakdown of the grinding organ that they have to break down those cells, it's age-associated, and so do aging interventions even then at a cellular level work to prolong the animal's resilience against that bacteria? And it is a really you hit on a nail on the head that I'm always wary of getting too defensive as a model-driven scientist. I think even the mice people get defensive about their work, is that no model system is gonna be perfect. And I've always thought of it in terms of, are you familiar with the Drake equation?
No, no.
It was the proposal for what's the likelihood that there's intelligent life in the universe? And so it's
Yeah, yeah.
yeah, yeah. So you multiply the number of stars by the number of
Mhm.
fraction that have planets, and so on and so forth. And so I think of drug discovery along the same lines. I think
Mhm.
that C. elegans and mice and even the other model systems people use, they're all just ways to improve our fraction of success. So, you know, I think the dream is AI could look at a million molecules in a heartbeat, give us 100 good hits. We could take those 100 hits and really rapidly look at them in Sealgans to find the 2 that we should spend our time and money on looking on in mice, and then, of course, the one of those that it works well in people. And so I see it as C. elegans are a useful tool in this ratchet system, but it's never the end.
Yep, yep, 100%. And I mean, I'm playing devil's advocate
You
in
should.
sake of
Yeah.
making a dialogue,
Yeah.
interesting, right? But I do think that that the approach that you guys are taking, it makes a lot of sense, right? I would say though, the thing that is the other common criticism that
Mhm.
you see about animal models, right, is like, well, maybe something just doesn't work in the animal model, even if it would work in
Yeah.
humans, right? But we just don't know, right? Like there are probably thousands of drugs that could have worked in humans that we just don't use or we don't investigate further because they happen to be toxic to mice, right? Or they happen to be toxic to other models.
Yeah. I think about, there's I don't know if you've, uh, highlighted in your podcast some of the great work about the dog aging project. Um,
Yet, not yet. But
cool.
I guess to reach out to Matt and or some of the other folks involved in the project. Yeah.
Oh, you and Matt should definitely do a podcast together. So the dog aging project is uh, it has the goal of using people's pet dogs to look at uh, aging over time in companion animals. The
Mhm.
hypothesis being that dogs are complex mammals like we are, and they share our living environments. So someone that maybe doesn't is super active is probably going to also have an active dog. Someone that isn't super active is going to maybe have a sedentary. Their dog is going to be exposed to more sedentary practices. So a lot of the comorbidities that we think of with healthy or unhealthy aging affect our companion dogs. It's a really, really cool system. But if I were to go and share a chocolate cake with my dog, he would have to go to the veterinarian in a heartbeat.
Mhm.
And I think that's like a foundational truth is that all these models are they're all flawed in their own way. I personally wouldn't take a drug that I've only seen C. elegans data for. It is part of the process, but it is not the process. And I think you raise good points. Like there's one of the strongest. This is data that we've published as part of that's actually publicly available on our website. Uh, we've tested rapamycin, one of the gold standards in terms of the current generation of longevity therapeutics, at in so many ways in C. elegans in our lab, and it doesn't really extend lifespan at the doses that are biologically relevant. It's yeah,
Seriously.
it's true. And so one of the things that we found with it is that the doses of rapamycin that are biologically effective, that are published in the literature as working in C. elegans, the chemistry of the rapamycin molecule is utterly insoluble at the concentrations that are reported. And so when we test out those concentrations, you see crystals of rapamycin on the Petri plate the animals are crawling over. And so they're incredibly, incredibly saturated with the drug, and you'll see a lifespan extension. But it is so far out of the context of a controlled experiment. Now, that doesn't mean that
Yeah. So
It's so weird,
tell
and
me
it's
that's the case. Like, how could that be possible, right?
right, that
Uh,
all this time we've been talking about it and going back and forth, and, you know, again, you can get a lifespan curve with rapamycin in the animals, it's just the necessary dosage is so high, it is crashing out of solution.
yeah.
Now, what's really cool is that then you can go and test other mTOR inhibitors and see we've seen ones that have, up to 50% or even more extension of animal lifespan. And so what that tells me is that, we've got the underlying conservation of the mTOR system, which people have looked at ad infinitum in terms of the mechanisms of TOR signaling. And it's conserved between humans and worms. But it lets us look for the next best thing that actually works in the same pathways as rapamycin, but at a lower effective dose.
Yeah, yeah. Maybe before we get into that, right? Because that is I think a little bit more complicated, and not a lot of people will be familiar with it. But and I wonder, maybe the reason, or maybe based on what you said, right? Like if rapamycin, at the doses in which it would work, it wouldn't be recommendable in humans, for example, because it would have like I don't know immune system, inhibition, for example, right? Like it would only work in lab animals, I guess, because they don't, they don't die of all the infections that we would find, going out in the street or whatever, right? Like, I mean, I guess, I don't know, like maybe the controls on those experiments were just not valid enough, right? Like, but this is one of those things that a lot of people would believe, right? And I guess it just does not reproduce.
Yeah, and I do want to put in a caveat. I don't want to say that I'm anti rapamycin. I work across the hall from Matt Caberline, and I'd find my office on fire tomorrow morning if I said that. I actually think that there's really good data showing rapamycin does work in humans, but it also works across a relatively narrow dosage and a relatively specific dosing regimen. And even then, just like you said, there's side effects. So, talking to people that have taken it, uh, you end up with mouth sores
Yeah.
and a host of other things that it's maybe it's worth it for the impact across other parts of your health and longevity, but I still think it's
Maybe it's not placebo control, right? There's no control against, good diet and exercise or whatever, right?
yeah, exactly,
Uh,
and that's where I do like to sit, I there is a lot of folks on both sides of rapamycin that are working really, really hard to understand it. I like to sit a little bit on the sidelines personally and wait for the science to be done on it rather than, uh, uh, throw shade at either party because there is so much of that right now in the longevity space.
yeah, yeah. Fair enough. Yeah, I guess people kind of like to argue a lot with
It's
these.
true, yeah.
But
Yeah.
before going into that, right? Like so you mentioned one thing that is very interesting, and I don't know if everybody familiar with, which is this notion of evolutionarily conserved, right?
Mm.
Which I guess if I may and do a little bit of a brief introduction, right? I guess we have um sequence, right? Like the genome sequence of lots of different animals, right? So we know that a lot of sequences they kind of appear many different times over time, right? Like we know some animals are kind of evolutionarily older, right? Like they appeared
Mhm.
earlier on Earth, and some of them appear later, right? And we also know that some mechanisms seem to be the same across many different types of animals, right? So we mentioned the mTOR, which is nutrient sensing, right?
Mhm.
This this mechanism that detects when a cell has food, right? And then it kind of uh metabolizes, I guess, that food, and then it creates proteins, right? And this seems to be repeated across many different pieces, different species, right? So that's why we kind of suspect that if it works in worms and in mice and in other animals, right? Like probably it may work in humans too. Like, I guess that's the basis, right? For trying to kind of look at this mechanism as opposed
Yeah,
to something else, right?
exactly, that's a great way to put it, and, we could even I love the idea that you put on there in terms of evolutionary time, because people use that to our advantage. Some of the work that I was doing under Dr. Patrick Phillips and University of Oregon was looking at different nematode species that the evolutionary goal and because these are old species, the evolutionary gulf between some of them is greater than the evolutionary gulf between a mouse and a human. And to be fair, they look like worms and the worms look pretty similar, but there is this latent amount of drift in between them that you can use to understand how well things translate across large evolutionary and temporal gaps.
And maybe one last question, just to kind of close on that, right? Like, I guess, like, why don't we use monkeys, for example, right? Like, monkeys are a lot closer to humans, I guess. I don't know, like a few, is it like single digit millions of years, right? Uh
Yeah, I believe so, but I'd have to double check.
yeah.
Uh,
But why wouldn't you use monkeys? Because they are more expensive, or like they are harder to handle, or like, I mean, it's like you would imagine that the results would be like a lot more closer, right? Like, they even like in many ways, like they have, social contracts like we do, right? That behave, similar to how humans may behave in some occasions and so on.
yeah, no, and I think that the two main reasons for that are uh, ethics and expense. And so, with doing primate work, you end up with a huge amount of regulations that are there for a reason. These are animals that are socially complex, and you want to make sure that you're doing the necessary experiments and you're doing them in the right way that is as humane as you can be. Um, it's one of the reasons that I think people are drawn to invertebrate work and work on simpler systems is you get to do any kind of science without having to worry as much or focus on trying to mitigate this makes me sound like a mad scientist, but ethical responsibility. The worm with 300 neurons doesn't really feel pain. We can do experiments and give them drugs that we don't know the cause and effect for yet and see results without subjecting an animal to undue suffering. And I think that's a really strong point for it. The other is that it's expensive. All of that infrastructure takes a ton of money and time and person hours to keep going. And with that, the monkeys actually live a long time as well. And so I will complain about the lifetime of an aged mouse being 3 years and being that a long time to do research. Monkeys live a whole lot longer. And so right now there is some of the great work that folks have been doing looking at, first it was caloric restriction in non-human primates, and now even some early data on long-term rapamycin effects showing beneficial outcomes is being published now, but the system is incredibly slow. And that's the gears churn slowly.
Yeah, yeah. No, fair enough, right? And I guess, also playing a little of devil's advocate again, right? Like, we as a society have decided that it's okay to have, 1,000 pigs in a building, for example, right? But we decided it's not okay to have 1,000 monkeys. The pigs are just as smart or smarter than monkeys, right? From what I understand.
Yeah. Yeah,
Uh,
it is a lot of those lines are arbitrarily drawn. And I think, we also don't talk about the amount of human suffering that goes into these
mhm.
experiments. You know, I was gonna uh, you know, the amount of suffering that graduate students across this globe as they're toiling away late hours, keeping the mice or keeping the monkeys alive and things like that for like next to no pay. And we kind of said that that's okay and that suffering is fine, just like we say that the mouse getting cancer is part of the process. And I love that you're bringing up some of those ethics because it does kind of flip the script a little bit, and I think asks some challenging questions of us as scientists and researchers.
Yeah, yeah. Um, maybe going back to works, right? So like I think in my mind, right, like as an engineer, as
Mhm.
somebody that comes from the AI side of the world as opposed to the biology, like the thing that you said that really resonates is the fact that the worm lives 20 days, right, or a month or something like that.
Mhm.
So your iteration speed is much faster, right? So if you have
Exactly.
to try one thing, wait 3 years, try another thing, wait 3 years, right? Then, like you're going to move very, very, very slow, right? Like in computer science, I guess, we we kind of compile our programs and then run them, and then we check things every few seconds, right? Like even when you build a product and you put it in front of people, and then you learn things, and then the next week you're releasing something else, the next week you're releasing something else, right? So I think this idea of, well, can we get a model that is useful, and then the iteration cycle is 3 weeks, right, or 4 weeks, and so, yeah, instead of 4 years, right? Like that makes a lot of sense. Uh, so so what you have done, I guess, at Aurora is basically enable people to do that, right? Enable that,
Mhm.
super, super fast iteration cycle.
Yeah, that's our goal, and I like the way you put that. I actually one of the reasons I'm where I am today is 'cause, I love answering biological questions. I am a little impatient, and I like this isn't 100% true, but I often find that when it comes to writing software, the answer isn't necessarily, "Can we do this?" It's, "How long will it take to get working?" And I love that idea, and I think that the closest we can get to that with biology is often using these simple species to get a quick yes or no that lets us iterate and move forward.
Yep. Yeah, I mean, in my mind, the way that I think of, drug discovery in particular, right, is you have all this essentially infinite number of options, right? Like people, give random numbers, right, that are
Yeah.
beyond, the number of photons in the universe. Or to me, that makes no sense. And I think it's better to think about it as it's essentially an infinite number of options. Like if we think about a protein, for example, right, instead of a molecule, because we can count proteins, I guess, easier, right?
Mhm.
So like you have 20 amino acids, right? And you have proteins with like, I don't know, I guess, hundreds, thousands of different, base pairs, right? So if you take, 20 to the power of 1,000, that is a lot of options, essentially infinite, right? So you have this essentially infinite search space where you want to, quickly, validate things. Uh, and uh, and in my mind, what is really important is you have some kind of heuristic that makes sense, right? And then how fast can you actually do it? I think the main problem in drug discovery is not so much that things fail, right? It's the fact that they take forever to succeed or fail, and they cost a lot of money. When they succeed or fail, right? So like if we can drive down those two things, right? Like even if the probability of success is the same, you could say 20% probability of success, but you don't need to spend 10 years and $2 billion, right? Like people normally quote. You just say, yeah, you know, can we do it in 1 month, right? For $1 million, right? And suddenly just have all this, shots on goal, right?
I love the phrase "shots on goal." That's how I think about it day to day, is that, there's so much unexplored space out there, and, we keep on developing things that look a lot like metformin or rapamycin, and a lot of the training data sets have metformin and rapamycin as the things that work. And so we need to actually go beyond that. And I think that's going to be kind of the next generation of therapeutics are going to be AI derived molecules tested really rapidly with, I'd
Mhm.
love it if it was our system. I love in vitro's robotic laboratory system. Like those tools are going to let us actually accelerate things much, much faster.
Okay. So let's talk a little bit more about that, right? Like, so we know, maybe for people that don't know about it, right? Like rapamycin inhibits this nutrient synthetic system, and I guess metformin, that's what does metformin do? It was something. I don't remember what was the mechanism of action of it.
It works across, let's say AMPK. But let me actually double check that and let's edit that.
Yeah, yeah.
Okay, so
Yeah, I think I just remember like it's some kind of partial inhibitor of let's say mitochondrial complex 2, something like that. So it kind of
yeah,
makes the cell use less energy.
okay, yeah, and I did a quick chat GPT to check myself, and it does also hit AMPK. That'd be a, if I got that wrong, Matt would just he would be out there and pissed off, and he wouldn't know why. He just know that a graduate student of his was wrong.
Yeah, well, I'll make sure to send him the episode once it's out there.
Well,
And
did me thinking about it.
um, so yeah, I mean, but I think those two are where I'm going with this, right? Is those two are, I guess, small molecules, right?
Mhm.
Well, I mean, I guess uh, rapamycin is not that small, but it still kind of counts as a small
Yeah.
molecule, right? So these are things that you just put in solution and then put in I guess in a petri dish, where the worms are doing their thing.
Mhm.
But what else could we think about in terms of longevity, right? Especially things that we could test in mice, right? Like and like what I'm getting with this is there's all sorts of gene therapies, for example, that are cell therapies that are kind of DNA damage repair mechanisms or whatever, right? Like that we can do that. Yeah, would be a lot harder to test, right? Because you can't just put that therapy right in a petri dish, right? Like you would need some kind of vector and so on and so forth, right? So like what kinds of things can we do with worms, for example, and what are the kinds of things that we would have to do with with other models?
Models, yeah. No, it's worms, while they do have a huge suite of genetic tools available for looking at uh, genetically, genetically modified proteins or labeled proteins or even knock down whole gene pathways. We can do that in a heartbeat in worms. But I do think that
Like
worms are
cool,
unique.
right? Like not live
Yeah,
from them. Oh, okay, interesting.
yeah, yeah. So worms have this really interesting evolutionarily derived system where RNAi is a system shared by a lot of animals, where small RNA transcripts direct gene silencing or gene modification. C. elegans actually can take exogenous, so RNAi from outside their body via their food, and their mechanisms will latch onto that and then silence those genes. So you can feed them a slightly modified strain of bacteria, which is really easy to make, and then you can knock down a gene pathway like that.
Oh, interesting. And like we humans, we digest it, right? So it doesn't work
Yeah.
for that. Yeah.
We can't do, unfortunately, we don't have the system to do oral RNAi. I wish. It'd be great for gene therapies. Um, I mean, like, this is actually, I think I'm gonna have a little bit of a hot take on this, is that I'm less excited about gene therapies. I think they are really useful in a really targeted way, but I think the future of longevity medicine in the near future, talking the next 10 to 20 years, is gonna be small molecules. If we think about the amount of good we can do with a therapy that targets underlying biology aging, be it for a disease endpoint or even for just improving healthy aging, small molecules are much easier to get to patients across a wide range of income brackets, a wide range of, uh, socioeconomic classes, and can be made. Yeah, I don't want to say take it for granted that they all can be made more affordably. Like we've seen some of uh, the dangers with off-use labels around, GLP-1 inhibitors spiking the price for people that wanted to use it for its prescribed use for diabetes, right? But uh, that being said, I think that there's a lot greater chance that we can get stuff to a much wider array of people with realistic technology in the next 10 years compared to gene therapy.
Yeah.
I still am excited about gene therapy, but I think that in the terms of longevity, the future history of longevity therapeutics, small molecules now are the steps that are letting us run so that gene therapy can let us fly down the road.
Yeah, yeah, and I completely agree, right? And I think this is interesting, right? Because a few weeks ago I had somebody on the podcast, right, and we were talking about DNA repair
Mhm.
mechanisms and mechanisms to protect the integrity, and I thought, well, I mean, this is great, right? And like I think like cancer being a somehow a stochastic kind of disease, the longer you live, the higher the probability of developing cancer. Like there's no way around it. That basic mathematics, right, versus the statistics, right? The more cells you have, the longer they live, the higher the probability. So at some point, if we say, well, maybe to live beyond 100 150 to 100 years, right, like you're going to have to solve those mechanisms, right? Like you're going to have to have genes that protect DNA replication integrity better than what we have right now. Like other species that live those 200
Mhm.
or 200 and change years, right? I think I mentioned the bowhead whale, right? That lives, I think, about a couple of hundred years and definitely has more cells than we do, right? So they have these better mechanisms. But if I think of putting myself in the shoes of an FDA, I guess inspector and say, well, you come here and you say this therapy, you know, when you take it for the next 20 years, or you take it once now, and then 60 years from now, 60 years from now,
Yeah.
50 years from now, it will avoid. It will reduce the probability of you developing cancer. And you want to give this gene therapy that changes every cell in the human body to somebody who is healthy. right? As a kind of like a prophylactic, you'd be like, you need to just get back home, buddy. Like you have no idea how any of this stuff is going to work, right? Like I would imagine it's impossible to get any of that stuff approved, like now or like anywhere in the foreseeable future.
Yeah, so I actually don't think I've had a chance to tell you about some of the work we're doing with the Air Force, have I?
No, I don't think so.
Yeah, so you hit on a great point that the one of the barriers right now to a lot of good uh that longevity therapeutics can do are those regulatory roadblocks. And in particular, you know, a lot of us in the longevity space are taking things that we know work on longevity influencing pathways and we're taking it and saying, okay, let's use this for cardiovascular disease or cancer or, you know, pivot and let's go and try and see if it does anything to Alzheimer's or Parkinson's. We I'm really proud of this idea that Aura had. So there's been some really interesting work looking at the effects of space travel on astronauts. There's been some great twin studies looking at how I think it was Mark Kelly and his twin, the different gene expression in space versus on Earth. And so, you know, one of the factors and a major contributing factor is the amount of stellar radiation that they experience.
Mhm.
That isn't
Not gonna
necessary.
believe this, but I was literally talking about that with a previous guest in
Oh, good.
podcast. Yeah, so you're going to see this is very funny. No, go ahead, continue.
No, no. Normal listeners, this is your space radiation drinking game. This is your shot here to take a shot since we've brought it up. So we had the hypothesis that if we took therapies that we found that work on longevity in worms, and we subjected those worms to radiation, those therapies would be protective against it. And we were able to work with it was a SBIR grant, which I don't know if you've talked about the SBIR mechanism on your podcast yet.
I don't think we have, but like I guess that's another kind of worms, right? Like that's exciting right now. Yes.
Another can of worms is a great way to put it. I'll just say, for those of you that out there that haven't gone through that, it's one of the major non-dilutive funding sources that startup companies can get from the federal government. So every government agency does them. We were lucky enough. We worked with the Air Force and Space Force last year to run this pilot project, and right now we're looking at working with our stakeholders there to advance it in the future. While we had originally thought about it just in the context of astronauts, one of the things that we realized was that there's a need for this not just for astronauts and people, you know, on the space station, but if you look at the radiation exposure rates for pilots, flight crews, they're actually significantly higher than folks that work terrestrially, let's
Mm.
say. And so, one of the things that we're looking at is developing longevity therapies for, you know, initially it'll be, you know, acute and chronic exposure to radiation among service members, but eventually then extending it outside of that to enrich for people that are uh, at risk of the symptoms of DNA damage. So we liked for the scientists, we like to say molecular sunblock, right? A longevity intervention that upregulates your resilience against environmental stress. The folks on the Air Force side that are excited about jets, uh, they've told us they like to call it molecular countermeasures. You know, the flares that Tom Cruise spits off to get the missiles away.
Yes.
And so that's kind of, I think, gets at your point is that there's these novel avenues that longevity biotechnology and biotech as a market are really exploring. And I don't think we yet know how the next generation of interventions are going to get to patients. And so we've got to take all these shots on goal.
Yeah. Yeah. I mean, I think this is one of those things that is inarguable, right? Like if instead of trying one thing, you try 10 across a different spectrum, you will have a higher probability of success. There's just no way around it. Um, I'm personally partial. I always say, with a caveat that I'm actually an investor in the company, right? But I'm partial to what the folks at Repair Bio are doing. They
Mhm.
basically created a cell therapy, right? That I would say, yeah, I don't know if I would call it the cell therapy or more of a protein therapy. The key of it is this protein that helps dissolve 7-ketocholesterol, right? Which is one of the things that accumulates in the arteries, and then ends up causing cardiovascular disease, right? But I really like the idea of removing damage, right? I think
Yeah.
that is nice because the kind of the logical rationale for it makes a lot of sense to me, right? Like people accumulate certain types of damage, right, in the cells. If you can remove that damage, it stands to reason that people would live long. But I don't know how somebody could argue against that. But then, if it's safe enough, then I guess you could apply it to a lot of other folks, right?
Right.
And I guess in some way, people have been trying to do this for Alzheimer, right? In many ways. So like we don't really know how Alzheimer works. The only thing we know is that amyloid betas and misfolded tau proteins are damaged, right? So we try to remove them, and then we'll see what happens, right? And I think there's a couple of approved antibodies, right, for that now, but still, it's kind of really unclear, right? But I mean, I don't know. So I think that approach makes a lot of sense. And the reason that I really like it is precisely because of this iteration cycle, right? So if you try to prevent damage from happening, for like, 50 years, right? Like it's a lot harder to prove than if you can remove that damage, in one shot.
Yeah, I mean, I think the ability to repair it is also really interesting. So we actually, with our system, one of the things we iterated on is looking at the difference between if we gave the animals the drugs ahead of exposure to, in this case, we used ultraviolet radiation as a really basic radiation source. Uh, we looked at what happened if we give the animals the protective molecules beforehand versus what if we expose them to radiation, then looked at the effects later on. And we still can see some animals have a greater chance of restoration of function even when given the treatment after the acute exposure. So I love one of the what you're talking about, what that company is doing.
Yeah, yeah. Um,
Can I ask you asked me so many great questions. Can I ask how on the AI side, so the work that you're doing, I think you I hope the people on your podcast get a chance to ask you about it, so you get a chance to talk about it. I'd like to pick your brain and hear how you're working to revolutionize more of
yeah.
the drug
So
discovery via AI space.
yeah, I mean, we haven't really shared much about that yet because we're still working on our V1. Even as of today, right? Like, so we started working with a few kind of early stage design partners. Haven't talked much about it, but I think the key idea, there's maybe a couple of interesting ideas that we're trying to focus on. One of them is in silico methods can be useful, right? Can be predictive of some things, right? I think they're not perfect, right? The same way that models are not perfect. The same way that even like not everybody responds the same way to a drug, right? Like so even in humans, right? Like sometimes things work, sometimes they don't. But I think the notion is that they are predictive of certain things, right? Like so, in many ways, you can predict toxicity, or you can kind of estimate efficacy, right, using simulations. And the notion is the better those simulations, the more predictive those models would be. Which again is a little bit of it's not for certain, right? It's a little bit of hypothesis, right? But in many cases, I think it's possible to do that. I think in some areas is more clear than others. So like I was talking with John Stokes, for example, is doing research on antibiotics, and he said for antibiotics, it just hasn't worked very well because nobody has done the right simulations, right? So people have tried a million different things, right? But at the end, you just kind of have to test in vivo. I would say in other areas, people have used rational drug design, and there are drugs that were discovered, using those rational drug design methods, and that has worked out well. Um, if you look at I don't know, like some cancer therapies, for example, like CAR T-cell therapies or
Mhm.
whatever, right? Like we would take uh the genome of a cancer cell and figure out the toll-like receptors, or like other kind of cell markers, and then we find antibodies that match those, and then put them in cells or whatever. Like the whole process is very rational in a way, right? It's from my understanding of it. So anyway, the long story is, we think those in silico methods can be useful, but the problem is they are hard and they take a long time, right? So, the one thing that we have right now is we have ways to automate, those
Yeah.
methods, language models and other things. So the idea is, instead of having a whole team of chemists, a whole team of medicinal chemists or scientists, going and creating the simulations, running them, iterating, understanding them, and so on and so forth, can we just use language models to roll stuff automatically, right? So, you know, in some ways, it's something that you can't really argue, right? Like if you believe that in silico methods work at all, if you can do the exact same thing but faster and cheaper because you have the computer do it for you, that's better,
Yeah.
right?
I love that. One of the things that you talk about taking shots on goal. I tried to get funding for this last year via an ARPA project. You know, Working like this, it's almost, like people talk about digital twins, right? Those are some of the tools that AI is letting us actually really run with to do really rapid prototyping and evolution, rapid testing in people. I wanted to make a nematological twin, and it did not get
Mhm.
funded. I wanted to make a digital drug discovery worm, and I couldn't get money for it. So I would like to work with you.
Let's do it. And I think, yeah, who is very interested in this kind of stuff as well.
Yeah.
The Chan Zuckerberg
Mhm.
initiative, they have this project to kind of create a virtual self. And I think they want to create a virtual self at a maybe molecular level, which I think is still a little bit hard. I think it will take many years. We don't know enough about that yet. But if you went and said, you know what, let's create a model at the pathway level, for example, right? Or even kind of mesoscale level, like different, cell organelles or whatever of all the cell. And, in a nematode, if you have only a few hundred cells and, like it may be a lot easier to create something like that from a perspective of, they have another model that's called, I think it's called transcriptomic perturbation model.
Mm.
We basically train it on transcriptomic data, and then you apply different perturbations and try to predict, well, what happens, right? If I inhibit this gene, what happens? If I, kind of, give it, something else that enhances expression of another gene, then how does that affect everything else? So I guess, that could be built for a nematode, I guess, and, would be a lot easier than trying to build that for a human.
I think so. I have a f I actually now I have a follow-up question. So we've talked a lot about the great things that AI can do for, biotech and longevity medicine. Do you have any downsides that as an expert in the field, you're worried about of using AI in the space?
Um, you know, so that's a hard question, right? I would say, the one thing that comes up all the time is the moment that you automate,
Mhm.
you kind of replace something that was done by a person, right? So, there's this underlying risk of like at what point do you use machines instead of using people. I don't think that's really going to be the case in science. I think it may happen in other areas. um yeah, I keep talking to people about marketing, right? Like, ChatGPT can write a marketing
Yeah.
email better than 99% of us can, right? So, if your job is writing marketing emails, maybe you should start, kind of looking at other things too. uh but science, there's just so much stuff that we don't know, right? So I would say, if anything, we will have more science,
Mhm.
when we have better tools. It's the same with software, right?
Yeah.
I think we've seen, maybe it's a little bit harder to get into the space if you're a software engineer, but there's still like millions of software engineers around the world, right? And what has changed is that, before you would spend a month to build some piece of software, and now maybe you spend 2 weeks, right? Because you can do it much faster. So I hope it will turn out, the same way. But yeah, I mean, I personally am not super scared about the things that people kind of raise up, like, folks being able to create megaviruses and like, so stuff, yeah, I think it's, you know, I, it's still, hard enough to do things like that, but and I'm not super scared about biosecurity, and so I don't know. Are you scared about those kinds of issues?
Biosecurity, not really. I think, the beauty of I kinda alluded to earlier, biology it is chaotic systems and it's complicated. And, uh I think it's hard enough to get biology to do something right, that getting biology to do something wrong is also gonna be incredibly hard. I'm less worried about that. I still think it's always good, like, obviously we need mechanisms in place to supervise that and make sure that no one's doing anything harmful, but I am less scared about the capacity for AI to accelerate those wacky things. I use AI every single day. Like, not only Aura has a AI-driven pipeline starting from our work back when we were still at the University of Washington that analyzes all of our data for us in an automated fashion. Uh, but I'm a CTO of a small startup. It would be I should be fired if I wasn't having a ChatGPT tab open at all times.
Mhm.
I don't own any s I'm not getting paid by them to say this, but, like, it's helped me submit twice as many grants in this last year and take those shots on goal and helped me we were successful in getting 2 SBIRs last year, which I would love it if it was more, but that still was really good and helped us grow our team and advance some of these projects, and
That's amazing. Yeah.
it it ChatGPT was, influential in the writing of those and getting them going.
Of course.
ChatGPT, I'm a biologist by training that loves writing code. I use ChatGPT to help make up for the fact I don't have a, computer science degree daily.
Yeah. Yeah. And I think also like in many ways, you're using technology to replace incredibly manual work, right? Like, if you needed to have a person looking at a petri dish where a worm is moving, right? Like, that experiment could never be done, right? Like it would cost, thousands and thousands of dollars, right? Or like,
Yep.
many people, right? Uh, so I think the competition is not necessarily a person, right? Like the competition is basically like, it just would not be done. Yeah, a
Yeah,
friend of mine.
yeah. I was gonna say, Javier, I was listening to some of your older podcasts, and I heard some of the shots fired at the Caberlin lab at the yeast dissection. I actually put that on speakerphone around the office today. Um, but you weren't. I hope you know. I'm joking. I totally understand where you're coming from, and it's the point of why we build these tools is to automate it. For those of you that aren't, uh, do you have a fan name like Polyphonetics?
We don't have any ad yet.
Okay,
Yeah, no, listeners, I guess, whomever
listeners,
listening
your
to
goal, your homework is to come up with a fandom name for Javier and his team. So, you know, it is like science has been incredibly tedious and it's been hamstrung by the need to do things manually. That's why we built the Wormbot that lets us do things much, much faster. You know, I think that, from my perspective, so l- slight tangent, not that big of a tangent. One of the things I'm so sorry, Javier. You're trying to run a
No,
podcast.
this is great. I think people will actually enjoy it very much. So this is what people like, right? This is,
Yeah,
honest conversation.
I I I I am really passionate about science education. So I'm a strong believer that, we are going to fail if we don't raise the next generation of scientists. So one of the things I like to do is I go and I give talks at high schools. One of the kids that worked with me and came in on the weekends while I was in lab, he got a first place prize for his science fair project at the Washington State Science Fair.
Yeah.
And what automated tools have me and our team do is, yes, it's replaced having someone coming in and actually poking the animals every single day, but it's meant that I've been able to ship data to students all over the country to, get them in for a quick set up an experiment, look at their data, run an analysis, run a trial, and actually expose them to the side of science that is, I think, most exciting, which is making a hypothesis, running a test, getting your data. And so I think that like these automated tools are going to, you know, there will be some jobs that don't exist anymore, but there is going to be a lot more access to science for people that are interested in having a hypothesis.
Yeah. Yeah. I mean, I love that, right? And I'm thinking, right, like, maybe, I don't know, this is a little bit, I imagine, alien for a lot of people that don't understand how science is done, right? Like, but I guess, uh, maybe, check if this analogy is valid, right? It's a little bit like an architect, right? An architect goes and draws a building, and then you need a couple hundred people to go and build that. But yeah, you need the people to get the steel beams in place, and people to draw all the soil, and then put every single brick, and
Mhm.
and install the plumbing and the electrical and so on and so forth. But at the end of the day, like a single person, right? Like an architect is the one that has the vision, and then puts it, recreates in 3D, creates the final renders, and then you say, well, this is the building, right? But then it takes an incredible amount of effort, right? So I guess we need more of those scientists that can act like architects and come up with smart hypotheses and experiments. And, maybe we just need a lot less, builders like putting one brick after another, after another, after another. But like that work still has to be done, right? But maybe
Yeah.
you can have a computer doing it, right?
I think so. And, not to shamelessly plug the Million Molecule Challenge, but our we started a grassroots open science project focused on letting people from anywhere over the world basically pay for the cost of reagents, and we've been running experiments for them for, I think we're just about 18 months in right now. Uh, People have been it's been incredible seeing people from all over test random ideas that some of them I think are incredibly smart. We've found some things on there that people proposed that extended worm lifespan by 50%, which is as good as like anything in the field, right?
Yeah, tenda, la carta.
We make anything that goes through the Million Molecule Challenge is 100% open access. We make sure of that. We've also, I can highlight our good friend and partner Krister Kauppi at Rapamycin Longevity Labs, who we've worked with to raise a crowd-funded project where we're testing about 500 not like mTOR inhibitors that have not necessarily been looked at all for their effects on longevity, and we're going to make that data open access.
Yeah.
Um, yeah, and so it is incredibly rewarding to get to work with people that are science minded from all over the world and work with their hypotheses. It is like having 1,000 professors though, which is incredible, and also some of them have my personal phone number now, and they know who they are.
Oh goodness. Yeah, no, that I can imagine that's not that much fun. It's like, I mean, it was checking another, completely unrelated to this, but it was like this system that this person that I know was building, and I was like, yeah, well, I guess I have your email, and then I'll just start filing my bug reports on Twitter or via email, and now like you won't be able to get rid of me, right?
Right, yeah.
Um,
I mean, it's so it's I wanna be super it's so worth it, and I would never have done this differently, and please, no one at me.
and I mean, I think we're already over an hour. The time really flew by, but I think we should probably close it around here, right? Because otherwise, people don't listen to the podcast, right? Like, I think some people put up this multi-hour long podcast, but then it's too much, especially for like interesting and intellectually stimulating and challenging things that we're talking about today, right? So I don't know, like maybe to close with, right? Like, if somebody has any question, about you or whatever, like, can they reach
Mhm.
out to you, I guess, on Twitter or LinkedIn or something like that, or how would somebody ask questions if they're interested? How would you?
Yeah, I'd love to. Thank you for plugging that, Javier. So, if you'd like to ask me any questions, feel free to Twitter, LinkedIn are both great. I think the easiest way is just ben@aurabiomedical.com. Send me an email, let me know what you're thinking. We also have contact us forms that we check regularly on our website. And, please be in touch. We're just excited to talk about the science we do. If anyone has any questions, please.
Awesome. Well, it was a pleasure. I enjoyed this very much. So thank you so much, Ben. It was a great conversation. So I hope everybody else that is listening, enjoyed it as well.
Likewise, and thank you so much for having me on. And I'm going to workshop a name for your fans as well. I count myself one of them. Javi Maniacs.
Oh my goodness. Yeah, let's punt that to the next one.
All right. Shots on goals.
Exactly.
All right.
Thank you so much, man.
Take care, Javier.